students, we were discussing chapter number 8, cell, the basic unit of life. In this chapter we have done the introduction of a cell, and we have also seen the difference between the prokaryotic cell and the eukaryotic cell in the last session. Now we have to understand the basic structure of each individual type of a cell, means prokaryotic cell and the eukaryotic cell, and we will start one by one. So let's start first with the structure of a prokaryotic cell. We will discuss the structure of prokaryotic cell with more emphasis on a bacterial cell. Okay, so let's start with the structure of a prokaryotic cell. Prokaryotic cell, like the other cell, it contains the three components. The first component is called as cell envelope. This is the first component that you can find into any cell. The second component you can find in any cell is a nucleus. And the third component you can find in any cell, it's a cytoplasm. So like the other cell, the prokaryotic cell or the bacterial cell possesses all these three: cell envelope, nucleus and cytoplasm. So let's start a drawing of this prokaryotic cell. Schematic drawing of a prokaryotic cell. So if you see that this is a prokaryotic cell, okay, this cell is covered by a boundary. This is a boundary. This boundary is called as cell envelope. What it is called as? Cell envelope. Okay. Now the second component that is present is a nucleus. Okay. So here this cell, it contains a nucleus, and nucleus contains a genetic material called as DNA. But as it is a prokaryotic cell, this nucleus is not covered by nuclear membrane. The nucleus is even not well organized, okay, and it is not covered by any kind of a membrane, that is a nuclear membrane. So here there is no membrane, okay, and this DNA that is present into a nucleoid region. So this component, okay, this is called as a nucleus. And what this nucleus contains? The nucleus contains a genomic DNA. What it contains? Genomic DNA. This genomic DNA is responsible for genotypic characteristic to a bacteria, or it gives a genotypic characteristic to a bacteria. Okay, so that is about a nucleus. Now, when we are discussing the genomic DNA that is responsible for hereditary characters, the bacterial cell also possesses a small circular DNA. A small circular DNA it possesses outside the genomic DNA. Outside, this is a genomic DNA. Outside the genomic DNA, it has small circular DNA. This small circular DNA, it's called as plasmid. And this plasmid is responsible for phenotype characteristic. Okay, it gives phenotypic characteristic to a bacteria. And one of the most important phenotypic characteristic it can give to a bacteria is resistant against antibiotic. I again repeat, one of the most important phenotypic character that is acquired by a plasmid is resistance against antibiotic. Means if it acquires resistance against antibiotic, the antibiotic doesn't kill such kind of a bacteria. Okay, and that is one of the biggest problem of 21st century. Okay, so ultimate point is that the nucleus contains a genomic DNA, and outside the genomic DNA it contains a small circular DNA called as plasmid, and that is responsible for phenotypic characteristic. So this is the second component, what we call it as nucleus. Now anything which is present between this nucleus and nucleus and cell envelope, this is called as cytoplasm. So anything which is present like this whole thing, okay, this is called as cytoplasm. Okay, anything between a nucleus and membrane or cell envelope, that's called as a cytoplasm. So prokaryotic cell contains all things: cell envelope, nucleus and cytoplasm. Okay, now we have to discuss each component in detail. So first we start with a cell envelope. Okay, It's a cell envelope, means covering or a boundary that is surrounding to a cell. This is a cell envelope. It is a bacterial cell. So this bacterial cell has 3 layers around them. How many layers around them? 3 layers. Which are the 3 layers around them in the bacterial cell? The first one, it's called as glycocalyx. Okay, first one is called as a glycocalyx. The second one, it's called as cell wall. And the third layer, this is called as cell membrane. These are the 3 layers which form a cell envelope. These 3 layers together form cell envelope. Okay, so let's see. The first one, the outermost layer is called as glycocalyx. So this is the outermost layer. Okay, this outermost layer is called as glycocalyx. The outermost layer you can find into a bacterial cell. Now the middle layer, I am drawing the middle layer. This is a middle layer. Okay, middle layer you find into a bacterial cell. What it can be called? This middle layer is called as cell wall. It's called as cell wall. So this is the second layer. You can call it as cell wall, the middle layer. Okay, so this middle layer, and the last, the innermost layer. This is the innermost layer you can find into a bacterial cell. And this innermost layer, it's called as cell membrane. Okay, this is the innermost layer, and this innermost layer, it's called as cell membrane, which can also be called as plasma membrane. It can also be called as plasma membrane. Clear? So how many layers surrounding or that form a cell envelope? 3 layers constitute together, it form a cell envelope. The outermost is glycocalyx. You can see this is the outermost glycocalyx. The middle one, it's called as cell wall, red color. And the innermost, this is called as cell membrane. Each layer has its own function. Okay, the usual function of this cell envelope is the protection of bacterial cell or protection of a prokaryotic cell. But let's discuss the individual function of each. The glycocalyx can be arranged in a 2 way. The glycocalyx can be either arranged loose, there is a loose arrangement, or it's a tight and tough arrangement. If it is a loose arrangement, then it can also be called as slime layer. What it can be called? Slime layer. If it is a rough and tough, or you can say tight and tough, then this layer can be called as capsule. It can be called as capsule. Okay. So the glycocalyx, the function of it is to provide the protection. It can be arranged in a 2 way. Either it's a loose arrangement or it's a tight and tough arrangement. If it is a loose arrangement, then it's called as slime layer. If it is a tight and tough arrangement, then it's called as a capsule. Okay, that provides a protection. Now the middle layer, this is called as a middle layer. Which is a middle layer? Cell wall. The function of this cell wall, this is a cell wall. The function of this cell wall, it gives a shape to a cell. The shape of a bacterial cell is due to a cell wall. And the another function of it is also responsible for a protection, and it prevents bursting of a cell. It prevents bursting of a cell. So this is a function of cell wall. So what is the function of cell wall? The function of cell wall, that is, it provides or it gives a shape to a cell, bacterial cell, and it prevents a bursting of a cell. Okay, so this is the function of a second layer. Now the third layer is called as cell membrane. This is the innermost layer. The characteristic of this innermost layer is such that this is a cell membrane. The characteristic of this innermost layer, it is a like other cell or eukaryotic cell, it is semi-permeable and it is selective permeable membrane. Okay, that means it will only allow a selected molecule to cross it. Any molecule cannot travel through it. Only selective molecule can travel through it. and it is a semi-permeable. That means it can also maintain the pressure, osmotic pressure equal on both the sides. Okay, so it is a semi-permeable and selective permeable membrane. Okay, and that is the same as you can find into other types of a cell, including eukaryotic cell. Okay, so in short, there are three layers which constitute together form cell envelope: glycocalyx, cell wall, and cell membrane. The glycocalyx may be loose or it may be tight and tough. If it is a loose, then it's called as slime layer. If it is a tight and tough, then it's called as capsule. Okay, the middle layer is cell wall. It is responsible for providing the shape of a cell, and it prevents a bursting of a cell, and thus it protects a cell. The third one is cell membrane. The cell membrane is similar to other kind of a cells. It is a selective cell permeable and semi-permeable membrane. Okay, so this is about the characteristics of the three layers you can find in a bacterial cell. Okay, now apart from this, what it contains? Here the cell wall, the composition of cell wall may vary from bacteria to bacteria. Okay, and based on the composition, the bacterial cell can be of two types. I am writing over here. The bacterial cell can be two types based on the type of cell wall it has. Okay, so which are the two types? The first type, it's called as gram-positive bacteria. Gram-positive bacteria. And the another one, it's called as gram-negative bacteria. Okay, how many types of bacteria? Based on composition of a cell. Based on composition of a cell, there are two categories of bacteria. First is gram-positive bacteria, and the second one is gram-negative bacteria. What are called gram-positive bacteria? Gram-positive bacteria are the bacteria those who can take up the gram stain. Those who take up the gram stain, or they can be stained with a gram stain, they are called as gram-positive. And if they don't stain, or if they don't take up the gram stain, then they are called as gram-negative bacteria. Okay, so what is a gram stain? Gram stain is a chemical or a stain, coloring agent developed by a scientist, a gram. So this is called as a gram stain. And upon the contribution of this gram, the bacteria can be named either gram-positive or gram-negative. In short, if they take up the gram stain, then they are called as gram-positive. If they don't take up the gram stain, then they are called as gram-negative. Okay, and staining is due to differences in the structure of cell wall of bacteria. All bacteria doesn't have a same kind of a cell wall structure. They have a different cell wall structure, and based on that, these are the two categories of bacteria: gram-positive and gram-negative. Okay, now the another thing. Next, another thing, you can see that like there is also a presence of certain infoldings, like you can see here. These are the infoldings, they generate from the inner layer, cell membrane. Cell membrane is the innermost layer. From this innermost layer, there is a formation of infoldings. Okay, these infoldings are called as, what they are called? These infoldings are called as mesozoa. What they are called? Mesozoa. The infoldings are called as mesosomes. This mesosome can be in the form of tubule, like tube-like structure. It may be in the form of vesicles, vesicle or bag-like structure, or it may be in the form of lamellae. Okay, so this mesosome can be in the form of tubules, vesicle, or the lamellae. Okay, so this is what is a mesosome. Mesosomes are the specialized infolding they generate from innermost layer cell membrane, and this is the characteristic of prokaryotic cell. The mesosomes are not found into a eukaryotic cell. Mesosomes are found into a prokaryotic cell. Okay, so this infolding may be of different shape. It may be a tube-like called as tubule. it may be a bag-like called as vesicle, or it may be a flat structure called as a lamella. Okay, so in any form it may occur, and this mesosome has important functions. So what are the important functions of this mesosome? The function of this mesosome, it is responsible for cell wall formation. Okay, this is the most important function. Through this mesosome, its outer layer, cell wall, the red color, that is going to be formed. Apart from that, they also have a role in DNA replication. It has a role in DNA replication. Okay. Now, apart from that, it also has a role in respiration. It also has a role in secretion. So these are the functions of mesosome. These are the functions of mesosome. Clear? I again repeat, what are the mesosomes? Mesosomes are the infoldings generated from cell membrane. It is the unique characteristic you can find into a bacterial cell, which can be in the form of tubule, vesicle, lamellae. It performs this function. The important function, it is involved in cell wall formation. It also involved in DNA replication. It is involved in respiration, and it's involved in a secretion process. Okay, so these are the 4 important functions in which they are involved. Okay, now some bacteria, like I am specifically writing the name of this bacteria. See here, I am writing over here. Okay, some bacteria, the name of a bacteria is a cyanobacteria. Some bacteria, they possess specialized foldings that generates from a cell membrane, and this is called as chromatophore. What it has? Chromatophore. And this chromatophore contains a pigments. What it contains? It contains pigments. And due to these pigments, they can do a photosynthesis. Okay? Usually, you know that bacteria may be a saprophytic, or you can say in general they are the heterotrophic. Okay, but some bacteria like a cyanobacteria, which can also be called as photosynthetic bacteria, they are autotrophic because they contain a specialized structure generated from cell membrane. What is the name of this specialized structure? It is called chromatophore, and this chromatophore contains pigments like chlorophyll. Okay, chlorophyll like pigments, and hence they can prepare their own food. Hence these bacteria are autotrophic in nature. Okay, they are autotrophic in nature. So students, we have discussed the first 2 components of bacterial cell. The one is a nucleus, that means a genomic DNA, and a small circular DNA plasmid. Second, we have also discussed the cell envelope. Okay, and we will discuss the other components into a next session. Okay, till then, students, keep learning, keep watching, like and subscribe this channel. Thank you for watching.